This is Tao Unbound, the English language podcast of Tel Aviv University. Please welcome your host, Ido Aharoni, Tel Aviv University's graduate, member of the Board of Governors, lecturer, writer, and veteran diplomat.
Welcome to another episode of Tao Unbound. I'm Ido Aharoni, your host, and it is my great privilege today to welcome to our studio Professor Ben Maoz.
Yep.
Welcome, Ben, to our studio. We're so happy to have you. Professor Ben Maoz is a professor in the Department of Biomedical Engineering at the Sagol School of Neuroscience, and we will talk about his scientific work, but as we always do, we begin with a set of personal questions. So, I read about your science, and it's incredibly sophisticated, and I'd love to learn more, and I want our audience to learn more. But first, tell us, when did you discover that spark?
So, first of all, thank you for having me today. Today I had actually two meetings with, let's say, the young generation. And I told them that it doesn't matter what you do, as long as you have a spark, you should follow that spark and you will be good at it. And for me, I loved science from day one. I remember that as a kid, I tried to mix different materials and to see what happened. I remember that I heard in the news that there is a problem with the environment and that carbon dioxide is a problem. So I went to my grandmother's car and tried to manipulate different things. And I loved science. I really loved science.
Where were you raised? What part of Israel are you from?
So I'm from Herzliya originally. And again, throughout my younger age, or let's say, high school and not elementary school, but mainly high school, I went to biology, physics, chemistry, all these things. I was really with short patience. So I didn't go to all the lectures and everything because I didn't have the patience to sit down and to study. But specifically chemistry and biology, I really loved it. So I was eager to go to the lessons. And I remember that basically I became a really good friend of the teachers. And they gave me the opportunity to do experiments by myself. And one time my teacher, she gave me like all the things that were needed for the experiment. And she went out and I wanted to be extra safe. So I made a pile for all the waste that I'm doing the experiment, and all the chemicals that I need, that I finished with, I put in the waste. And apparently I didn't know that I shouldn't mix base and acid. And there was like a huge smoke that started to dissolve the pile. And she came in running. What the hell are you doing? What the hell are you doing? So, and I enjoyed it. And I felt, wow, this is what I want to do when I will be a grown-up. And I knew that I want to follow chemistry. Basically, I wasn't that good in school. My grades were so-so. And again, it was because I didn't put a lot of effort in it. I didn't really invested time and what is needed. And my parents were very supportive. They were all the time, told me, as long as you pass whatever you need, and we trust you. And I started with the 5 Yechidot, as we say in Hebrew, like the 5 grade,
like
the
highest level.
The highest level in math when I was in Yud, in the 3rd grade. Then I went to the 4th when I was in the 11th grade. And then I went down to the 3rd when I was in the 12th grade. Again, because I didn't come to the lessons. And I said, I'm not going to do my Bagrut, my finals, because I know that after the army, I'm going to redo it with better grades and the highest level I can. And my father told me, look, I never told you what to do, but here I'm telling you, you need to have a diploma at the end. Like, you want to do better afterwards, you should do it. So I basically sat down on my ass for 2 months, taking all the materials, and I went to the grade to the 3rd level in math, did it, and after the army I basically redid all the courses again because I needed to have good grades to get into the university. I knew that I want to learn chemistry. I did my bachelor degree in chemistry, master degree in chemistry, and PhD in chemistry. Because you know what they say, chemists have solutions, right? So I really loved it.
And what was your PhD about?
So basically, my PhD was, in science, we can give like really amazing, explosive titles for things that I worked on nanoplasmotics and chiral effects on nanostructures. At the end of the day, it was to use nanostructures to manipulate light to get extraordinary effects. What does it mean, extraordinary effects? It means that you can do a detection of single molecules using unique structures that you manipulate the light, and there is the interaction with light and structure.
If you can say a few words for the benefit of our listeners and viewers that don't know what nano is all about, what is the meaning of nano? It's the ability to re-engineer or reorganize it on the molecular level, right?
Yeah. So basically, the word nano for itself, it's 1/100000th of a meter. Like the word is like have 1 kilo, 1 centimeter.
So it's a unit. It's a unit.
Nano is a unit. I can have nanosecond, nanometer, nanogram. Like nano is just a unit. When you talk about nanotechnology, people talk about the technology that is the size of a nanometer or nanometer size. And nanometer size is basically, let's say, a 1,000th of a hair, the diameter of a hair. So it's very, very, very small. It's much smaller than a cell, for example. So when you talk about nanotechnology, you talk about technology that is really, really, really small. Now, why it's interesting, it is because what happened is that the physics that we know is completely different in the small scale. For example, what is the color of gold? I'm asking you.
Gold.
Gold, right? Like all of us, we know the color of gold is gold, right?
Yeah.
Now, if you take, let's say, a cube of gold and you start to cut it, what will happen? Gold is still gold. A rose is a rose is a rose is a rose, right? So a gold is a gold is a gold is a gold. And you cut it down smaller and smaller and smaller. And there is a specific size that when you go smaller than that size, the gold become blue, red, yellow, green. The gold itself, without any colors and manipulations, the gold itself become red or yellow or green or blue. It is because the physics that you know that is working perfectly on the large scale become very different in the small scale. And it's true for many properties. This is just one property of light. There are properties of electricity. There are properties of magnetism. There are different physical properties that behave completely different when they are small versus big. So it's actually very interesting to work in the small scale.
Now, is it all theoretical at this point?
No, not at all. Actually, these effects that you're familiar with, or that here I'm telling you about, today we can explain them. But actually, many, many years ago, people used them without knowing the effect. There is, they call it, if I'm not mistaken, Ligarius cup. It's a cup from the Roman period that if you take, you will see the cup itself is green. And when you shine light from the middle of the cup, you will see that the cup itself become from green to red. And apparently it is because the Romans, they put really, really tiny gold nanoparticles that actually when you come with the light, suddenly you see the gold as red. So we know these effects. Now we know also the theory, but now also we have the technical tools to work in such small scale. We have electron microscopes. We have a focused ion beam that can help us to curve, and all the effects that you have in the transistors of your computer. These are effects that you can work with only in this small scale.
Now, that work led you to the current work of your lab at Tel Aviv University. You're concentrating in 3 different areas. Tell us about them.
So I will take even a step back, and I will say that, at the beginning, I knew that I'm going for chemistry. I did my master's and my PhD in chemistry, and then in order to get a faculty position, you need to do a postdoc abroad. And I knew that my postdoc is going to be in chemistry. I already found different labs in Boston to do research in chemistry. But then, when I went to Boston, I didn't want to limit myself to a specific lab because my wife, she is also a researcher at Tel Aviv University. Her name is Yasmin Meroz. She was also my lab instructor. She gave me the lowest grade I ever got. So then I said, love will last, right? And we got married. So she got also a postdoc in Harvard, and I knew the area that we're going to be in. So I said, so that I will have a higher chances to find a lab, I opened myself to different areas. And I went to the labs in chemistry, which were amazing. But I also went to a lab that specializes in tissue engineering, which was a field that I never touched. I was not familiar with this. And I remember that I came to his lab, Professor Kit Parker, and they showed me suddenly cells and tissues and things that you can do with this. And I remember that my eyes opened. I had this spark that you talked about, and I said, this is what I want to do. And I did like a huge shift. It's a big shift to go from chemistry, materials, that all the day you work with materials and chemical hood and all these nanostructures and things you talk about, suddenly to work with living matter. And the example that I talk all the time about working with living matter versus non-living matter is the following example. So you know what it is to play with Lego, right? I take my pieces of Lego, I put them together, I go for lunch, I come back, they are the same place. To work with living matter, it is to actually manipulate the kids that build the Lego. The kids, they have their own will. You tell the kids, okay, sit down and build your Lego. You go for lunch. Nothing will guarantee, or actually I can guarantee they will not stay at the same place. And when you work with living matter, you need to know how to work with cells. The cells themselves, they have their own will, their own, behavior. And in order that the cells will do whatever you want, you need to learn how to manipulate them, figure of speech, so that they will do whatever you want and not necessarily what they want. And sometimes it coincides. So this is what I learned during my postdoc. In my postdoctoral studies, I actually did this switch into tissue engineering and neuroscience. And this is basically what I brought here to Tel Aviv, that I started my lab a few years ago, about 7 years ago. And now, as you said, I'm at the Department of Biomedical Engineering, Sagol School of Neuroscience. And recently, together with Uri Asheri and Yaki Yafi, we established the Dreamer Fischler Center for Regenerative Medicine. Currently in my lab, we have 3 different fields. We have personalized medicine, we work on integrated electronics, and we work on biohybrid sensors. Each field is very, very different. And if you'd like, I can elaborate on each.
Please, yes, that's why we're here.
Okay.
Inspire us.
I will do my best. So let's start with what does it mean to study human physiology? Okay, at the end of the day, I'm not a physician. So it's not that you come to my lab and I will start examining you. What we do, actually, we tackle a very, very severe problem that is related to personalized medicine and drug development. Now, if you look at drugs, developing a drug, by the way, you know how much it costs to build a hospital?
I have no idea.
So give a rough estimation.
I don't know, 300 million.
You are actually pretty close. 200 million. You see, you're pretty close. How much money does it cost to build or design or create a drug? One drug.
I have no idea.
Give a rough estimation.
$1 billion.
Again, you were pretty close. $2 billion.
2 billion.
With one drug, you can build 10 hospitals. Okay? The question is why it's so expensive, right? So creating a drug, it's a very long and tedious process. It takes between 10 to 20 years, and it costs roughly $2 billion. The problem is that the drug development process itself, it means that you start with many, many compounds, you try them on different tissues, then you go to animals. After you succeed with the animals, you go to human beings, and then you get your FDA-approved drug. The main problem is that between 60 to 90% of the drugs that successfully pass animal trials fail in human clinical trials. Okay, let's understand this thing for a second. Here I have an amazing drug. It can cure you from cancer. I tried this drug on a rodent. The rodent had cancer. Now he's going back to his family, happy and healthy. I give the same drug to a human being, between 60 to 90% that the drug will fail. Okay, and why is that? Because human and other animals are not the same. And I can give you another example. For example, do you own a dog? Do you have a dog?
I used to.
You used to.
He died, yeah.
Unfortunately, but this is how nature goes, right? And I can give you a very toxic material that you will give your dog and he will die. The name of this toxic material is chocolate, right? You can eat chocolate without any danger, right? How can it be? Both of us are animals, but our physiology is different. If you will give paracetamol like Acamol to a mouse, the mouse will die. We know it's not the case with in our system, right? So there are discrepancies between humans and other animals. And the problem is that imagine that you do all this process for a really, really long time. And at the end of the day, when you do this transition to humans, it fails. So you need a different model. You need alternative that will be a better prediction for how it will work on us. So what is the best model we can use to predict how drugs will affect human beings? Again, it's a question.
Oh.
It's not used that I'm tackling him.
No, it's
You
good.
see? I wanted at the end of this podcast, see how many times he laughed. You see? Every time I tackle him, he laughs, right?
I know, it's actually, I would like to be interviewed about science, which I know nothing about. So I would assume real humans.
You're right. Would you volunteer?
Depends
on what?
On the level of risk.
I'm telling you, I developed this amazing drug in the lab. Amazing. Believe me, the best drug ever. Okay? I'm telling you, here. Believe me. I swear in my mother, it will work. Would you try?
Well, if I'm desperate, I will.
I don't want you to be desperate. I want to do like a clinical trial.
Again, depends on the level of risk, I would. But if it's risky, I wouldn't.
Well, at the end of the day, you don't know if it's risky until it's too late, right?
Right.
So at the end of the day, it's true. We would like to try drugs on humans, but to do it in an ethical way. An ethical way. So what we're trying to do in the lab is basically to create human models that are not human beings. Okay? What does it mean? We're not creating little babies, and we're not creating these embryos that we're doing experiments on them. Not at all. What we do is we use tissue engineering to try to mimic human physiology to try drugs and different things on. What does it mean? It means that, for example, we take human cells, we create the organ functionality with these tissues. We take this small organ-like tissue, we put it in a Lego chip. It looks like a piece of plastic, like a USB stick, but from plastic. And then we call it, for example, organ on a chip. I know it's not clear at all, but we will do another round of explanation, and then it will be better.
No, I think what you're saying is that you're imitating the behavior, the physiology of a human being, and thus allowing you to perform trials or experiments on the tissue and receive results that are similar to what you would from a human being.
First of all, you're completely right. But since I know it sounds too abstract, let me give a better explanation. So, for example, what is the kidney? What is the functionality of the kidney? Again, it's a question.
A kidney, I guess, is the ultimate filter of our body.
Perfect. So the kidney is a filter. So imagine I will take human kidney cells and I will create a filter out of this, and I will put this biological filter inside something that looks like a piece of Lego. I will call it a kidney on a chip. What is the heart? The heart is a pump. So if I will take human heart cells, I will create a pump out of this. I will put it in a Lego thing. I will call it a heart on a chip. Now, why I say Lego all the time? Because at the end of the day, the Lego, you can click together. So imagine I can take the heart on a chip, the kidney on a chip, the gut on a chip, the brain on a chip, the liver on a chip, the whatever on a chip, and to start to link them together to create microphysiological systems. Now, since these in your body, the organs talk with each other. So like this, you can mimic the crosstalk between the organs and to see what is going on. Now, this system is very powerful because and other labs as well, we were able to show that if you take specific drugs, we can get the same results as we see in the clinic. So we get really, really, really good results in using these tools compared to clinical data that we know.
Now, how do they talk to each other? Which technology are you using to imitate, for example, the neurological system?
So the neurological system, we can mimic, or we can actually create what we call brain on a chip and brain organoids and different type of neurons and subtypes of neurons, the sympathetic and the parasympathetic and central nervous system and things like this. But how your brain is actually talking to your lungs and heart.
It gives them orders, right?
How? Let's talk, for example, not the brain. Let's talk about the kidney, how or the pancreas. How the pancreas can talk with the rest of the body.
I don't know.
With the blood.
With the blood. Through the blood.
It releases different signals, and the insulin that's being secreted suddenly arrives to different organs. So we do the same. We have this blood-like material that flows between our different organs, and this is how they communicate. So this is what we do in the lab, and we work with many pharma companies and many different organizations and different labs to develop these tools. And each lab has different, research question, organs that they are interested in, and things like this. The thing is that this is not the end of the problems. We have many, many, many more problems. For example, we are not only very different from animals, but you are very different also from each other. Actually, at a given time, about 75% of the population receive drugs that are not optimal for their condition. Now, why is that? Because drugs are developed by looking at the population. Meaning now I have a drug, I will give it to you, to you, to you, to you, to you. People in China, people in India, people here in the Middle East, in the US, South America, Europe, wherever you want. And we look at the population.
And there's this assumed average.
Indeed. And I can guarantee that people from India and people from the US are not necessarily, they have the same physiology as you. The problem is even more severe because half of the population are actually not similar with, actually they have different organs, half of the population, female and male. We are different, okay? They secrete hormones, females, secrete hormones that we, males, do not have, and vice versa. They have estrogen, progesterone, we have testosterone. It's not the same. And in recent years, actually, there is a new field that is called gender-based medicine, because we see that there are drugs that has different optimum depending on the master-son cycle. So if you take it at the beginning of the month, the drug will be less effective than, for example, at the end of the month, and vice versa. So how can I say that a drug that was tested on a female will be good for me, and vice versa, in the same way? So what we're aiming is to do personalized medicine. And the way you can do personalized medicine is in the following way. We need to remember for a second how we came to the world and how we actually have tissues and organs. We have a sperm, we have an egg, they come together. This interaction creates a ball. This ball is a ball of stem cells. Stem cells can become any cell they want. Heart, brain, bone, muscle, gut, liver, whatever. And this creates us as a human being. We all know for many, many years that this is a one-way ticket, right? So your brain will not become a heart and your heart will not become a bone, right? Wrong. About 20 years ago, a professor in Japan named Shinya Yamanaka discovered a way to do this reverse. Meaning now we can take a sample from your skin or a sample from your blood and to transform the cells to be stem cells again. And then in the lab, we can actually transform your stem cells to whatever cell we want. We have this recipe. And I can take your own blood, create stem cell, and then create heart cells with your own DNA. Muscle, bone, whatever, with your own DNA. And this is the Dreamer-Fischler Center that we created here at Tel Aviv University that provides this also as a service. And we do service for labs, we do services for different universities, we do services for people that they want this, and also for drug companies. Now, why it's important? I will give you one example of something that it's not, science fiction. It's actually science that we do in the lab. The Jewish people, we were blessed with many things. One of them is many genetic disease. Okay, so we have, for example, here at Tel Aviv and Shiba and Ichilov, we have the largest cohort in the world of people with genetic mutations for Parkinson. What we do is we want to try to find the optimal drug for them. Instead of to go to a person who has a Parkinson disease and to tell them, can you give me a biopsy of your brain? What we do is actually to take a blood sample, and from this blood sample to create stem cells. These stem cells, they have their own DNA with the genetic mutation for Parkinson. We create mini brains with their own DNA and the mutation for Parkinson. And then we can try different drugs in the lab on these mini brains and to see which drug is the optimal for their condition. And this is only one example out of many. So the dream is basically to try and to use these tools as personalized medicine and to improve drug development. The last hurdle that is related to this project or this field that we're working on is regulation. Because at the end of the day, you need to have the regulation that is being set by the FDA. And until 2 years ago, this was a dream. 2 years ago, there was a change. The FDA changed the rule or the law that you need to do clinical trials, preclinical trials on animals before doing it on humans. And it changed it that you don't need anymore to do preclinical trials on animals before going to humans, but you can use these advanced models, such what I told you, instead of animals before going to humans. Now, it's relatively new. It's 2 years ago, but it did a revolution conceptually that now you can start to actually invest in these human relevant models as an alternative to animal models. So this is one aspect of what we do. When
is this going to be accessible to actual drug producers?
It is.
It is already.
It is. There are some companies that actually specializes in organs on a chip. We work with pharmaceutical companies, and pharmaceutical companies are starting to build departments specifically for advanced human models. It's like going on these days.
It's happening. So
it's happening. The revolution is here.
And you say that the trigger was the change in regulation 2 years ago.
The trigger, the trigger is a very delicate word in the sense that the trigger was that the technology was mature enough for the people who changed the law, for the regulators, to do this. So eventually the trigger was that, yes, the FDA and the Senate created this, change in the law, but the trigger was that actually the technology is mature enough to show that it has these capabilities. So it's,
incredible. So we're soon we're running out of time, and I wanted to ask you about your plans for the future. So you described incredibly compelling vision for the future on personalized medicine and your ability to imitate human physiology in order to do clinical trials or preclinical trials. What's next for your lab?
So I will answer this question depending on how much time do we have. Because as I told you, our lab is very, very interdisciplinary. And this is actually only one aspect of the things that we do. It is so interdisciplinary that the two other projects are actually not even related to what we do. One project, and I will just mention it briefly, is that we were able to restore tactile sensation. We created a device that you can actually implant at the tip of the finger, and it can restore the sensation for people who lost their ability to sense. This actually was a project that we started I went for a run with a good friend that he's a magician. He's such a good magician that he can actually take the fingers of people who lost their digits and to attach them back, and they can actually move their fingers again. He's the head of microsurgery unit in Shiba, Dr. Amir Arami. And he told me, look, about 70% of the people, I can restore their fingers and they can move their fingers, but they lose their tactile sensation. And we said, okay, challenge accepted. One thing led to another. We created this device. It's actually working very well. We tested it on different animals. Ramot, the company tech transfer of the university, helped us to establish a company for this. Company name is Tangible. And we were able to get about $5 million investment. And now this startup, we are heading for first in human in that sense. And this is, again, something really unrelated to this organs on a chip, but that has really significant clinical impact because, every time we have this, presentation for investors, they ask us, okay, who are your competitors? And it's the easiest question because we don't have any.
Right.
Currently, there is no technology for restoring tactile sensation.
The ability to restore sensation is only for the tips of the finger, or
no, not at all. It's true for any area of the body you'd like. We started with this because this was the more, most, the conceptually, the first thing we thought about because this is what he's working on. But now we're working with people with breasts that there was a breast reconstruction, people with diabetes that they don't feel they have diabetic foot, and other different things. So it's very modular.
Amazing.
Now, I told you that we have 3, and I told you out of 2. The last thing I will briefly mention, because I know that we are running out of time. Although she said we can chat how long that we want to, you see. So the last project we're working on is basically to integrate the amazing sensing capabilities of insects with robots. The humankind, we have amazing sensors. We have, amazing microphones. We have amazing cameras. But there are some sensors that we are really far behind what nature has. For example, the sense of smell. For example, the mosquito can sense the CO2 that we emit from a distance of 30 meters. And it is so sensitive that we don't have any good sensor that even equivalent for such high sensitivity. So we said, instead of competing, let's integrate. So conceptually, imagine that I can take the nose of the mosquito, integrate this with a robot, and then you will have a CO2 sniffing robot. And this is basically what we did. Instead of a mosquito, we used the locust, and it's a collaboration with Amir Ayali and Yossi Yovel, that we took the nose of the mosquito, we attached it into a robot, and now we have a robot that can sniff around, and with super high sensitivity. We are 10,000 times more sensitive, for example, for explosives, than the device that you have in the airport that touches your hand to see if you have explosives. We can smell whatever you want.
And so you could use those robots
For everything. Everything there is a smell, we can sense it. And this robot can go, is autonomous, so it can go in a room and to sense whatever you want, to sniff around. And, if you have something that you're interested in, so we can actually identify, and we use machine learning to see what kind of odorants you want to identify and to really identify them. Now, since I know we are running out of time, and you asked me how I predict the future in that sense, so I will need to quote Niels Bohr on that one. And Niels Bohr said that it's really hard to predict, especially when you talk about the future. And I can tell you that the future, look at this. If I told you like a few years ago, would you be like 2 years ago that, we need to be afraid of AI? You said, only in the Terminator movie, right? But look today with ChatGPT and all the different AI algorithms that today nobody knows what Google is anymore because the first thing you run into is ChatGPT. So things are changing so fast that it's really hard to predict. I can tell you that what we're aiming for, if you talk about personalized medicine, is that in the future, you need a liver donation. So if I will give you a different liver, your body will reject it because of the immune response. The future is you will give me a blood donation. I will create stem cells, make them a liver, 3D print a liver, and voila, I will give it back to you. I want to do personalized medicine. You give me a small blood, a vial of blood. I will make the relevant tissues, test them in the lab, and tell you, hey, this is the best drug you need. If we talk about integrated electronics, the technology improves all the time. You will have smaller, more sophisticated, different sensors. And when it comes to integrating, biohybrid robots, this is just the beginning. It's a really growing field, and the capability is basically, the sky's the limit. We can do many, many, many things. As long as there are problems, we have opportunity to find crazy solutions.
Well, thank you so much, Professor Ben Maoz. This was such a pleasure to be educated by you and to be inspired by you, because you gave us 3 different narratives, each with an endless horizon and endless possibilities. So thank you so much, and hopefully one day we'll host you again here to hear about your new developments.
Thank you very much. Thank you for inviting me. Thank you for the kind words. And I hope that the next one you will interview will make you laugh even more than what I did.
Yeah, well,
I just need to tell them the secret, to ask you questions.
Thank you for interviewing me a little bit. Yeah. Well, thank you again. And from Tel Aviv, until our next episode of Taiwan Bound, goodbye.